and welcome to BIO4YOU. Okay, in this video, we're going to learn about topic 7, recombinant DNA technology. Okay, let's check it out. Alright, so 7.1, Introduction to Recombinant DNA Technology. Okay, so here are the learning outcomes for 7.1. So we need to define recombinant DNA technology, list the tools used in recombinant DNA technology, and state their functions. So the glowfish is a genetically modified fluorescent zebrafish. Okay, this fish has been genetically modified. It became the first genetically modified animal to become so popular and open to the public as a pet. Means that the genetic of this fish has been modified to give it the fluorescent color. Originally, there is no zebrafish that is having a fluorescent color. Okay. So for the definitions, recombinant DNA means that it's a DNA molecule made in vitro, i.e. in the lab, with segments from different sources, i.e. different sources of DNA. Okay, if we put it in simple word, means recombinant DNA, we are joining several or a couple of DNA segments made in the lab. Recombinant DNA technology is a series of procedures that are used to join together, i.e. to recombine DNA segments of two or more different sources in vitro, i.e. again during in the lab. So here are the tools used in recombinant DNA technology. Okay, the first one is the target DNA or gene of interest. The second one is restriction enzymes, DNA cloning vector, host cell, modifying enzyme, or DNA ligase. We will look into a little bit of detail for each of tools. Okay, the first one, target DNA. What is target DNA? It is a specific DNA sequence which encodes for a protein, resulting in specific desired trait or characteristic which is not found earlier in a particular organism. For example, insulin gene, i.e. the gene will be inserted into the host cell, and for target DNA, we don't use the term desired gene because certain references or certain book, they use this term, so please avoid it. Okay, let me put it in simple word. Okay, what is target DNA? Okay, basically target DNA is a segment of DNA, is a part of DNA taken from some other sources and then will be put into the DNA of our host. Okay, so maksud dia macam ni. Contoh saya ada host. Contoh host tu adalah bakteria E. coli. So dalam bakteria E. coli ni tak ada genetik untuk insulin. But then we take the genetic for insulin from human cell and then we put it into the host cell, i.e. the bacteria just now, and the bacteria later will start to produce insulin for you. Okay, so that's the main purpose of one of the main purpose of this recombinant DNA technology. So that's why we need to use the target DNA, the DNA or the gene that we want to express, i.e. we want to show its trait. Okay. Alright, so this diagram shows you how we're going to use this gene of interest. Okay, so initially we have the bacteria. So what we have in this bacteria, we have the bacterial chromosome and we also have the plasmid. So what is plasmid? Okay, actually plasmid is also the bacterial chromosome, but the difference is that it is shorter and we usually manipulate this plasmid where we will take out this plasmid from the bacteria and then we're going to insert the gene of interest and later we're going to put this recombinant plasmid into the host cell. So we're going to take the gene of interest from the cell that contains the gene of interest. For example, if we want to have a human insulin, we're going to take the gene, that part of gene, from a human cell. Okay. The second one is restriction enzyme or restriction endonuclease. Okay, by definition, it is an enzyme that recognize, i.e. Be able to identify the specific base sequence of DNA and then cut the DNA at specific site, i.e. the restriction site, to produce DNA fragments. Okay, and these enzymes are isolated from bacteria. Okay, to put it in simple word, this restriction enzyme, um, the function is very specific. Certain restriction enzyme only recognize certain specific base sequences. Maknanya dia akan cari hanya dekat tempat yang spesifik tu saja. Dia hanya kenal dekat bahagian situ, and then dia akan potong, and then dia akan cut open the DNA. So that is the function of restriction enzyme. Why do we need a restriction enzyme? Because remember, we want to add something into our plasmid. So before we are able to put something, first you need to cut the plasmid to make an opening. So that is the function of this restriction enzyme. Once you have cut open the plasmid, then later you are able to put something or your gene of interest. So here are the example of restriction enzymes. Actually, there are many types of restriction enzymes, but the one that you need to study in our syllabus, there are two. Okay, the first one is EcoR1, that's how we pronounce it, and the other one is SMA1. So EcoR1, actually Eco stands for Escherichia coli, the name of the bacteria, and this one SMA1, SMA is taken from the name of the bacteria Serratia marcescens. Okay, so this EcoR1, it only recognize this base sequence GAATTC. Okay, it also reads the same for this one. Remember, we are only reading the base sequence from 5' to the 3' direction. We don't read this sequence as CTTAAG. No, we read this sequence also as GAATTC, because you always read from 5' to 3' direction. However, for SMA1, it only recognizes this sequence, which is CCCGGG. Okay. Okay, as I mentioned from the previous slide, different restriction enzyme recognize and cut different sequence of DNA, and different restriction enzyme cut at particular base sequences called restriction sites. Many of the base sequences recognized by these enzymes are palindromic. Okay, so what is palindromic? Okay, we look at the next slide. Okay, palindrome is a sequence in which the base pairs are read the same on complementary strands in opposite direction. Okay, it means here like this. So we have one, okay, we know that DNA comes in double-stranded, okay, so they are double-stranded. So this part of the DNA reads as GAATTC, and opposite dia pun from 5' to 3' also GAATTC, means reads the same. Itu maksud dia, reads the same. So for this one, CCCGGG, also same goes for this one, CCCGGG. Kan sama. So that is what is meant by the term palindrome. So restriction enzyme produce either blunt ends by SMA1 or sticky ends by EcoR1 after cutting at the restriction site. So when both strands of the DNA molecule are cut in the same position, no nucleotides are left unpaired. This is a blunt end. Okay, blunt ends means tumpul. What is meant tumpul means hujung dia sama sahaja. Okay, what is the meaning of sticky ends? We will refer on the next following slides. But we look at this diagram, so we see here that these enzymes of SMA1, it cuts at the palindromic sequence of CCCGGG, but the cutting was made between C and G. Maksudnya dia akan potong antara C dengan G. So same goes on the opposite strand. So it will also cut between C and G. That will gives you exactly a blunt end. Okay, so there are no nucleotides left unpaired. Maksudnya tak ada nucleotide yang tertinggal tidak dipairkan. Okay, by comparison, we will look on to the next slide for the comparison with sticky ends by EcoR1. Okay, So when each strand of the molecule is cut at a different position, so that one strand, the 5' or the 3', overhangs by several nucleotides. Overhangs ni maksud dia tergantung lah. These single strand ends can spontaneously base pair with each other. This is a sticky ends. So sticky ends is this part. Okay, so they are left overhang. Okay, so this is due to the eco R1. When it is making the cutting, it will cut between G and A of this strand, and it also makes the same cutting between G and A for this strand as well. So we see the G and A are now separated. The G and A are now separated. This separation will produce a sticky end. There is this TTAA, same goes with here, TTAA left unpaired. Dia tak ada pairing dekat sini. Yang G dengan C ni still pairing. Yang TTAA left unpaired, i.e. overhangs. So this is what we call as sticky ends. Just now, for blunt end, there is no overhang. Okay, so this diagram shows you the comparison side by side between sticky end and blunt end. So, yep, for eco R1, it will mix the cutting shown by this arrow between G and A, So, it will make this cut, and here are the dotted lines, then they will separate it, these dotted lines. For the blunt end, the small one will mix the cutting between C and G, here C and G, so it will gives you a blunt ends. There is no overhangs. Nucleotides over here produced. Okay, so this diagram also shows you the emphasizement of how the restriction enzyme makes its cutting, okay, and produce the sticky end. And later, okay, we have the another segment of DNA, i.e. from the gene of interest, alright, being inserted, and the gene of interest also need to have the same complement base pairing cut with the same enzyme. So you see that later it will complement with the sticky end, and it will bind nicely. Okay, so this DNA fragment produced by the same restriction enzyme, we have the AATT here binds with the AATT here, and this AATT here also will bind with the AATT for this one here as well. So finally, what we have, the recombinant DNA molecule nicely being combined with the plasmid and the gene of interest. Okay, next, a DNA cloning vector. So what is DNA cloning vector? It's a DNA molecule that can carry foreign DNA into a host cell and replicate there. Okay. So there are several types of vectors, and mostly these are the common vector that is being used, and these are the list of cloning vector that you need to learn and a little bit memorize them. Okay, so we have the first one, plasmid. Just now I've mentioned earlier during in this video, I've mentioned about plasmid. So plasmid, okay, it has many variants. Okay, the one that is commonly used is PAC18. Okay, and the capacity to carry insertion size is 15 kilobases. Okay, what does this mean? Okay, plasmid is only a small section of circular DNA found in the E. coli. Okay, since that plasmid is only small, having a short sequence, it can only carry small insertion size. What is insertion size? Okay, just now I mentioned about we can insert gene of interest, but how big the length of the gene of interest can be inserted? Maximum for plasmid is 15 kilobases. Kilobases means thousands, means 15,000 base pair can be inserted maximum into the plasmid. Okay. However, for bacteriophage of the strand lambda 2001, okay, 23 kilobases can be inserted. For cosmic, 44 kilobases, and we have the biggest here with the yeast artificial plasmid. Alright, so up to 600 kilobases. That's very long, quite long, okay, for the gene to be inserted. So usually, when we only use a short gene of interest, we will only use the closest possible vector. Okay, so for example, insulin is only a very short, having short base pairs. Okay, so we don't have to use we don't have to use cosmate. It is enough. It is sufficient if we just use plasmid. Okay, here is about Plasmid Pak 18. It is a small circular double-stranded DNA in bacteria, specifically E. coli, that can carry foreign DNA into a host cell and replicate there. Okay, replicate there is also important because you need to able to make many copies of the recombinant DNA. Okay, we use the term recombinant DNA to address atau untuk panggil DNA yang sudah been modified. Maksud modified ni apa? Dah ditambah dengan gene of interest. So kita panggil sebagai recombinant DNA. Different from the main bacterial chromosomes and replicates on its own. Okay, so maksudnya lainlah daripada main bacterial chromosome yang asal. Carry genes that often encode protein, i.e. enzyme, that protect the bacteria from antibiotics. For plasmid, it need to contain 3 components. First, dia mesti ada an origin of replication, and then it needs to have a polylinker to clone the gene of interest. This also called as multiple cloning site, where the restriction enzymes can cleave ataupun cut. It also have an antibiotic resistance genes, i.e. a selectable marker. So, what is origin of replication? So, ni dia ori. This is the point where we start the replication process, and then a polylinker called as multiple cloning site ataupun short form dia MCS, multiple cloning site. So, what is this MCS? Actually, along this segment of gene here, di sepanjang gene Ligase ni ada multiple cloning site, whereby in this segment we have many sites where a restriction enzyme possible to make its cleave, to make its cut. Okay, we see there's a list of names here, so I'm going to pick the one that you guys are familiar and you guys need to learn. Here, EcoR1. What is meant by this label here is that EcoR1 will exactly makes its cutting here. Whereas for small one will exactly mix its cutting here. So that's why it is called as MCS, multiple cloning site, because at this site it has multiple site for cleaving, for it to be cut. And the third one is antibiotic resistance gene. Okay, for antibiotic resistance genes, we're going to learn towards the end of the video. Okay, so however, this diagram also shows you this is the plasmid. Okay, for Pak 18. So here is the Ligase gene. We know that along this Ligase gene there is MCS, multiple cloning site. For here is EcoR1. Okay, and another restriction enzyme. Okay, and then DNA insert means that DNA can be inserted at this point here. So here is what is meant by the antibiotic resistance, where we know in this part of the plasmid ada segmen yang dipanggil sebagai AMP-R. AMP-R ni, AMP stands for ampicillin. Ampicillin is the name of the antibiotic. R means resistance, means that this gene here is resistant to the antibiotic ampicillin. Maksudnya dengan ada genetik ini dekat sini, dia adalah kebal daripada antibiotik ampicillin. Okey, kenapa nak ada kena kebal daripada antibiotik ampicillin? We will discuss this on later slides. So here next is the characteristics of cloning vector. It is able to accept. It must be able to accept foreign DNA in multiple cloning sites, okay, able to replicate independently in the host cell, okay, having origin of replication initiation or ORI, possess selectable genetic marker, i.e. such as the resistance to antibiotics such as ampicillin resistance. Okay, here is the characteristics of a host cell. So a host cell is an organism that had been used to receive a recombinant DNA for cloning purposes. Okay, remember again, recombinant DNA means having the DNA that have been modified, yang jadi alter. And it will host for the host cell for plasmid is genetically engineered E. coli. So plasmid biasanya kita akan masukkan dekat dalam E. coli. Okay, next is continuation for the characteristics of host cell. Apakah ciri-ciri host cell yang bolehkan dia dipilih digunakan untuk recombinant DNA teknologi ini? Able to receive DNA recombinant through the transformation process. Okay, nama proses untuk receive DNA ini adalah transformation. And able to maintain the structure of the DNA recombinant from one generation to generation. Okay, it is very logic. Of course, you need to have the host cell able to receive DNA recombinant. If the host cell cannot receive DNA recombinant, what's the purpose of it being a host cell? Okay, so a host cell also must be able to maintain the structure of the DNA that is being received. If the host cell only able to receive and then later it will damage or attack or destroy the DNA recombinant, again that host cell does not fit for its job. Okay, and next one is able to amplify the gene product from the DNA recombinant. Means that the gene of interest that we insert earlier later will produce its product. But if the host cell cannot amplify, tak boleh nak memperbanyakkan lagi product of the gene of interest, again that host cell is not fit for its function, hence we don't choose that cell as a host cell. So we are only going to choose a host cell that is able to amplify the gene product. So most commonly used host cell is again E. coli for the plasmid. So host cell need to be relatively larger than recombinant plasmid. Of course, you need to have a ample space to do the process, and you also need to have the antibiotic resistance. Okay, last but not least, for the tools is the modifying enzyme, which is DNA ligase. So for DNA ligase, it is an enzyme that catalyzes joining of DNA fragments permanently by catalyzing the formation of phosphodiester bonds between sugar phosphate backbones. And this is very essential for DNA replication. Okay, remember earlier when I mentioned to you, we have the plasmid. So with this plasmid, how we going to insert gene of interest? Of course, first we need to cleave the plasmid using restriction enzyme. So once the restriction enzyme has cut the plasmid at any particular site of the MCS, then we have the plasmid open up, and then is able to receive the gene of interest. And then what enables the gene of interest to be ligated or to be joined with the plasmid? You need this DNA ligase for this purpose. Okay, so this DNA ligase will join these DNA fragments by making the phosphodiester bond. Okay, that will be all for this part 1 of the video for chapter 7. Alright, so please stay tuned, and again, please like, share this video. Alright, see you on the next video. Thank you.